Hi, this is the continuation of the DNA replication lecture. Now we're going to talk, now that we have realized and been shown proof how that DNA replication occurred in a semi-conservative fashion using the old parent strand as a template. The next question became, well, how is this semi-conservative DNA replication possible? AKA, how are the DNA strands divided? How does the DNA replication occur? So if we pretend that this is a teeny tiny chromosome, hopefully you can see that these are two pipe cleaners twisted together. Does the whatever replicates the DNA attach itself to one end and just kind of unwind the DNA molecule from here, breaking the base pairs? Does it do from both ends, or does it do it from the middle? So it comes in here somewhere in the middle, opens up it ever so slightly, and then proceeds unwinding the DNA. And then filling the DNA was, filling the parent strand was new daughter strand that's being replicated. And when people looked at electron micrographs at replicating DNA molecules that they had stopped in action using various inhibitors, they saw these really interesting bubbles that would form on a DNA strand if DNA polymerase was allowed to have its way. You can see here that the bubble is a lot bigger and here it's a little bit smaller. And these places were then called origins of replication. And hopefully you can appreciate here that on one DNA molecule there's multiple origins of replication. So then the question becomes, how are the strands separated and how are the new bases added into the strand? So the hypothesis was that the bubbles represent the two single strands that have been separated and the new DNA has been added on the side, being the red base pairing was blue, and as DNA replication proceeded, the bubbles became bigger and bigger and bigger until in the end the bubbles merged and two DNA molecules were made from the original blue. And this was, people addressed that were able then to ask exactly in which orientation the replication is occurring by using high and low radioactivity level, by using thymidine of high and low radioactivity levels. So a low radioactivity thymidine will make a pale band on a film, which is not as black as the much more highly labeled high radioactivity thymidine. And as you can see here, since they could visualize the actual DNA strands on an electron microscope image, you can see that when they did the experiment, the original pulse was of high, and then the original pulse of the DNA was at of low thymidine, and you can see how it's a little bit more pale. And then the second pulse was of high activity thymidine, and you can see on this micrograph that it's a little bit more dark, showing that the DNA replication began here somewhere in the middle and then proceeded towards the edges of the circle. And you guys can see here that this picture is just flat out wrong. So they drew these bands here incorrectly. They're missing this whole another mirror image of this first low and then higher activity thymidine incorporation of DNA. So why is it that bacteria can get away with one replication origin? Hopefully you guys remember from Intro to Microbiology that bacteria have a circular chromosome and they are replicated by this rolling circle replication, but suddenly a second circle sort of looks like a figure, like an 8 for y. So bacteria have one origin of replication, and the eukaryotes have very, very many. They have one origin of replication every 10 to 40,000 base pairs, both in yeast, which is a primitive eukaryote, and in humans. And the reason for that is because eukaryotes have a lot more DNA than bacteria do. We have a lot of regulatory sequences, a lot of random transposons, we have all these promoters, all these interests, so much DNA. And if the eukaryotes would use only one origin of replication per chromosome, they would take more than 100 hours to replicate DNA of an average human cell. Because again, we're working with 10 to the 9th base pairs that the DNA polymerase has to replicate. As fast as it goes, it can't go fast enough if it's only 2 of them that are participating in a reaction. Now, if you have multiple replications origins, you can put 2, and in fact, there's 2 here and there's 2 here, DNA polymerases crunching along for each origin of replications. There's 4 polymerases per origin of replication. So a lot more polymerases can participate in the reaction, and things can get on a lot faster. So that an average eukaryotic cell still takes quite a bit of time to replicate. I don't remember how fast yeast replicates, sorry, but an average human cell grown in a lab, for example, HeLa cells, take about 24 hours to completely replicate their genome and then undergo division. And 24 hours is better than 100 hours that they would be stuck with if they only had one origin of replication per chromosome. Now, how is DNA replication initiated? So, the eukaryotic origins of replications have a charming name, Rs, courtesy of the yeast community. And it's a particular nucleotide sequence that the ORC hexamer, so these brown things are different proteins that make up the origin of replication complex hexamer. So the ORC will recognize the Rs. Once the ORC has bound, it will recruit 2 additional regulatory proteins. They don't have strong functions, but they recruit others to the location. So it recruits CDC6 and CDT1. You guys don't need to remember the fact that there's CDT1 and CDT6. But hopefully you will be able to remember that the next step initiation of eukaryotic DNA replication is the recruitment of this lovely hexameric molecule. You guys can see this in blue. It has 6 subunits. It's called MCM 2.7 in yeast, and it's the putative helicase. Now, if you guys remember me talking about motor proteins 2 lectures ago, helicases are the proteins that are necessary in unwinding the DNA. It is the helicase that answers the question of how semi-conservative replication can be done. How can we break the hydrogen bonds between 2 inside a double-stranded helix? It is the helicase. The ORC will come in and kind of open up the structure of your little chromosome here that has a bond. Then the 2 helicases will come, and then they're the ones who will start breaking the bonds and unwinding the DNA in order for the DNA polymerase, for the replication complex, to enter into the picture. So once the MCM27 has been recruited to an origin of replication area, or Rs, the cell is now ready to enter S phase. This whole recruitment occurs in G1 phases. All of the origin of replications, and there's, as I said, quite a few per chromosome, get occupied by the ORC and the helicases, and only after all of the origin of replications are occupied, a signal is given to the cell that it can enter S phase, and it can now start DNA replication. And that occurs during this time between G1 in G1 to S phase transition inside cell cycle, the pre-RC complex leaves overwhelmingly, aside from MCM27, which is the helicase. They're kicked out there by CDKs, which are a very important gene that regulate our cell cycle and that I'm not going to talk about, and recruits this whole massive shebang of the eukaryotic replisome. You can see how many proteins are involved here. And this, you guys, you will have to know. I don't care whether you know what's polymerase epsilon or polymerase delta, but you need to know, and I will have a slide later, about different functions that are required for a DNA replication to occur. There's many proteins with different functions that have to work in a particular sequence. And you guys need to know what all it takes. So once the pre-RC complex leaves, the helicase stays and everybody else is recruited. And now DNA replication can proceed, and that occurs in S phase. All right, and since usually in biology you don't get to see this complexity. For now, I'm just going to put blobs over here, and you guys can see that the DNA polymerase complex has arrived, and there's this massive DNA polymerase complex sitting here and here on the two sides of the opening replication bubble. So there's one here and there's one here. And when I say DNA polymerase here, there's actually two DNA polymerases, one doing the liking strand and one doing the leading strand. We'll talk about it. So, in fact, we'll talk about it now. The fact that the existence of the replication forks raised a new question of how DNA replication mechanism could possibly work, because it was realized pretty early on that DNA polymerases, aka the enzymes that synthesize the DNA, only work 5' to 3'. They cannot then synthesize the DNA in another direction. Yet the replication fork was clearly moving in both directions. So because the DNA is anti-parallel, you have a 3' to 5' strand here, and here you can start synthesizing DNA in the direction, here you can start synthesizing the DNA in the direction you like. You started with 5' somewhere here, and you're synthesizing it in this direction. So the direction of replication fork movement will be down this way. But this strand is going to have trouble because it's oriented incorrectly. So in order to accommodate for the directionality of the DNA and the anti-parallel nature of the double helix, the following solution is used by cells. Again, this is something you have probably already covered in integer microbiology, the Okazaki fragments, etc. To accommodate the 5' to 3' orientation and the anti-parallel nature of the double helix, the DNA replication is semi-discontinuous. The DNA replication is occurring. During DNA replication, there is such a concept as a leading strand, where the DNA polymerase can just crunch along because it's going in the correct orientation, 5' to 3'. And the idea of a lagging strand, where the DNA has to be synthesized in short chunks, the Okazaki fragments, and then ligated together to form a continuous stretch of new DNA. Now, you cannot tell apart after the DNA replication is done with which DNA strand was new and which one old. The semi-discontinuous nature of DNA replication does not leave any scars on the DNA. So, you guys can see that on the leading strand there is one DNA polymerase, and here I actually drew two, but there's actually only one that plays a role, usually. So question becomes, oops, sorry. How is it that how is the DNA on the lagging strand synthesized? On the leading strand, so another quirkiness of DNA polymerases is that they, certain exceptions aside, cannot synthesize DNA de novo. They need a primer. They need a small stretch of something that they can then elongate at very high speed and create DNA based on the parent strand template. So on the lagging strand, on the leading strand, there was at some point an RNA primer too. And on the lagging strand, there's a bunch of little RNA primers that are about 10 to 13 nucleotides long that allow, that are first put in by an enzyme called a primase, and then the RNA polymerase comes here and elongates from the primer, elongates the DNA into the 5' to 3' direction until it hits another RNA primer. So let's go through this process step by step, because it is a bit clunky, and hopefully I'm not repeating something that you guys have already heard a million times. So let's zoom out on this area. We have a lagging DNA template here in blue, light blue here, sort of pale blue right there. And we have the Okazaki fragment, the old Okazaki fragment that has already been synthesized, say right here, and the old RNA fragment. And now we're trying to make a new Okazaki fragment. And again, just to remind you, because the DNA orientation is anti-parallel, the bottom strand is 5' to 3', and the new strand is 3' to 5'. So the first thing that will come in is DNA primase. It is also a DNA polymerase. It's an exceptionally crappy one that likes to make mistakes and only does a few nucleotides at a time. As I said, RNA primers are about 10 to 13 nucleotides long. It will come to the lagging DNA template strand and write out a little primer and then leave. So the purple thing here is the little RNA primer that was synthesized by primase. Very easy to remember name, primer primase. Now, the next thing that will come in is DNA polymerase. DNA polymerase works 3' to 5'. So this little primer here is 5' to 3', so it can. So DNA polymerases works 5' to 3'. This little primer is 5' to 3', so it will bind right here and extend this primer, RNA primer, as proper DNA. And these stretches that the DNA polymerase made are called the Okazaki fragments, based on the discoverer of this DNA replication mechanism. Here goes DNA polymerase. It leaves as well. Now you still have the problem with this old primer, which is RNA and not DNA. So how does the cell solve that? There will be here, you will have, there's different other DNA polymerases that have 5' to 3' exonuclease activity. So they are not just synthesized DNA, but they can remove the old RNA primer in front of them. So they will be using the orange strand, the orange Okazaki fragment that was just synthesized as the primer, and then extend. And as they extend, they will excise the orange primer. You can see that the orange primer was removed, the Okazaki fragment became longer, and now there's just a small nick in the fully elongated DNA molecule, fully elongated DNA fragment. There's one covalent bond, one phosphodiester bond missing between this Okazaki fragment, the old one, and the new Okazaki fragment. So here go. There's a nick between the phosphate and the OH, and that has to be repaired. because DNA polymerase cannot synthesize that particular reaction. It can attach 5' to 3', but here we have, it would have to grab on to the 5' and catalyze a connection between the 3' OH here and the 5' phosphate here, and it cannot do that. A different enzyme called the ligase, which will, it's a more of a medical term, it will glue together two DNA strands, it will ligate them. It can synthesize the 5' phosphate to 3' OH linkage. It will come here, it will fix the nick in the DNA strands, and then it will leave. And now we have a perfect continuous DNA molecule in this stretch of the lagging strand. The process will then repeat itself over here and continue further. So, summarizing this little bit clunky animation is this slide showing you that the first step is DNA primase. It will come and make the primer. Then DNA polymerase. In bacteria, it's pol 3. In eukaryotes, it's pol delta. Again, don't need to remember that if you don't particularly want to. We'll extend this. I will not test you guys on the names of the actual enzymes. I will test you on the names of what a helicase does and what a primase does and what a ligase does, because that's important. The names of the actual proteins will change based on the species. Escherichia coli will have one name, and Bacillus subtilis will have a different name, and yeast will have a third name, and humans will have a slightly different fourth name. Okay, so step 1, primase. Step 2, polymerase. Step 3, a different type of polymerase, which will kick out the RNA primer and replace it with DNA. And step 4, ligase, which will seal the nick and make the two Okazaki fragments connect. Now that we have looked at how the lagging strand is amplified, let's look at how the DNA polymerase actually does its job. But first, I will take a break so I can download this lecture for you guys. So hang on.